From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and concerned citizens making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Assistant Professor in Pollinator Health in the Department of Horticulture.
The namesake of this podcast, pollination, seems like a pretty straightforward process that everybody knows about. You have these pollinating insects that go from flower to flower, transferring pollen, resulting in fertilized ovules that either, increase the number of seeds or allow a fruit to set. And in the process, when it comes to agriculture, results in increased yields and better tasting fruit and higher quality seeds. Now, this process is actually beset with a lot of complications, and you can quickly come up against this if you have a backyard. If you have backyard fruit trees, for example, that some fruit trees are self-incompatible. They need pollen from another variety to be able to set fruit. Now, this is well worked out in many orchard crops, but it isn't for a key crop in the Pacific Northwest. That's high bush blueberry, and that's where my next guest comes in. Dr. Stan Chabert is a postdoctoral fellow in the Mallinger Lab at the University of Florida, and he's part of a large U.S. Department of Agriculture grant that you heard about on a previous episode with Dr. Lisa DeVetter, working towards understanding the pollination systems of blueberry, trying to come up with a pollination planner, a way to kind of forecast the pollination needs for the present, but also the future. And a key part of that is knowing how different cultivars, what are their pollination requirements? And that's what Dr. Chabert is working on. In this episode, we're going to hear about his current underway research looking at different blueberry cultivars and their cross-compatibility. But we're going to transition towards the end of the interview and hear a little bit about his fascinating research doing his PhD work at the National Institute of Agricultural Research in France. So without further delay, blueberry pollination this week on Pollination. I'm so excited to have you on the show this week on Pollination, Dr. Chabert.
Hi, Anthony. Thank you to invited me. I am happy to be here.
I was excited to have you on because we were in a meeting, we're in a project together, and I'd seen some preliminary work that you had done on an ongoing research project on blueberry pollination. And maybe to start out, I wanted to ask a very basic question. In some crop plants, a fruit or a seed can't form unless pollen is transferred between different cultivars. And I'm sure anyone who grows orchard fruit knows that you have to plant a pollinizer cultivar within a row to act as a pollen source, or else you won't get fruit. What is the situation with blueberries? Do we need pollinizers to get blueberry fruit?
Yes, this is a good question because many growers, especially researchers, think that blueberry is not self-compatible. It is self-incompatible, which is actually wrong because blueberry and the whole Vaccinium genus is self-compatible. That is to say, when a pollen grain lands on a stigma, it can, if this pollen grain is self-pollen, so if this pollen comes from the same cultivar, this pollen grain can germinate on the stigma and can grow in pollen tubes through the stil and can fertilize ovules in the same level as for cross pollen, so pollen from another cultivar. So blueberry is self-compatible, but there is what we call early acting inbreeding depression. When ovules are fertilized by self-pollen, a part of these ovules are aborted.
Oh.
And so
let me get this straight. So it's self-compatible in the sense that if pollen from the same cultivar lands on the stigma, germinates down, it makes its way to the ovule, but in some cases it it doesn't result in a fully formed seed.
Exactly. It's only a part of these ovules, so a part of them they produce full seeds, mature seeds, and a part of them they just become small seeds. They are not well shaped, and we know that the size of the berries are linked to the number of the mature seeds. So if a berry have more mature seeds, so it will be heavier. And so that's why from the beginning of the 20th century, a researcher discovered this in 1920. So if a cultivar of a blueberry is cross-pollinated, so it will yield heavier berries. So a cultivar of blueberry doesn't need cross-pollination to set fruits, but cross-pollination can increase the weight of the berries, and so it can increase also the yields.
Okay, fascinating. So having more of those pollinated seeds can lead to larger berry size, which I imagine was what a grower is hoping to get at. Maximize the yield they're getting. Now, so this question of to what extent and which cultivars, are the degree of incompatibility is part of we're working together on this USDA project, and your part of the project is really focusing in on this question to test the degree of improvement in fruit set and berry weight that results from cross-pollination between cultivars. Can you describe the basic experimental setup that you are using to measure if and how much improvement cross-pollination brings?
Yes. So it is my current work with Rachel Mallinger at University of Florida as a postdoc. So for this part of the project, it was to know if some cultivars, because before this project, we thought that only a part of the cultivars, so northern or southern cultivars, only a part of them would need some cross-pollination to increase the berry weights.
Mm-hmm.
So the question was to test, to know what cultivars between northern and southern need cross-pollination to increase the weight. The berry weights, and so for this, I just worked in a greenhouse, and I choose some flowers on some branches, and I choose to pollinate only 3 flowers per branch, and on some branches I applied a self-pollination treatment with hand, and for other branches, I applied cross-pollination treatments, and I choose different pollen donors as cross-pollen donors to test if the pollen donor would have a different effect on the berry size and on other variables. And I pollinated only 3 flowers per branch because if we want to see if there is an effect of cross-pollination on the berry weights, we have to be sure that the branch or the berry is not resource limited. Because if we pollinate all the flowers on a branch, for instance, perhaps there will be not enough resources from the plants, and so all the fruits could set, all the flowers could set the fruits. But the effects of cross-pollination could be buffered by the lack of resources.
So you've got this greenhouse, you've set up these different cultivars, and you're doing 3 flowers per branch, because you really want to make sure that to see that berry size increase, because if you pollinated everything, then maybe they just didn't have enough nitrogen and the berry sizes are the same size, except they wouldn't have been if they had unlimited resources.
Exactly. Perhaps the berries will have more mature seeds because of cross-pollination, but there's more, this higher number of mature seeds will not be converted into higher weights because of a lack of resources.
I understand. Okay. What a great design. Okay. And so you had, how many cultivars you finished? I believe the southern high bush blueberry study this past year. How many cultivars were you looking at?
Yeah, I looked at 10,000 cultivars, and I will study 12 northern cultivars next year. And so this year I measured. So until now, we knew that so some of the cultivars would benefit from cross-pollination by increasing the fruit size and the time to ripen. But I also measured this year the firmness, the sugar and acid contents, and perhaps in the future we will also measure the anthocyanin contents to know if the cross-pollination could also increase some variables for fruit quality. And what I also discovered this year is that on my 10 cultivars that I tested, all of these cultivars finally benefited from cross-pollination for fruit size, time to ripen, and for the other variables.
All 10.
Sorry?
All 10 saw an improvement from cross-pollination.
All the 10.
Wow.
Before we thought that only a part of the cultivars benefited from cross-pollination, because I think in the studies the researcher didn't pay attention to the limitation of resources. So perhaps they pollinated. I don't know exactly, but perhaps for instance, they pollinated too many flowers, for instance, and so perhaps sometimes they didn't see the effect of cross-pollination.
Oh, because then in that case, there would have been a benefit, but the plants didn't have enough resources to really kind of result in a high quality fruit.
Exactly. And perhaps this is why some growers don't think that cross-pollination could increase their yields, because the plants don't have enough resources. And so all the flowers set fruits, or nearly all the flowers, but there is no increase in berry weight and yields because there is not enough resources in their field.
Can I ask one quick question? I think I've missed this. I should know the answer to this. But in terms of the pollen donor, so you've got these, you have a self flower that you self-pollinate, but also you have flowers that you pollinate with another pollen from another cultivar. What is the pollen source for this outcrossing? Is it a mixture of all the different cultivars or one specific?
So for self-pollination, I harvested pollen for the same cultivar, but on other plants, because there could have an effect. We are not sure, but if we collect the self-pollen on the same plants, perhaps it would have another effect, a less effect compared to other plants. And for cross-pollination, I choose different cross-pollen donors from several plants. And I also tried, this year, a mixture of these cross-pollen donors to see if there would be a synergy between the cross-pollen donors on the berry weight and on the other variables. But finally, I didn't find any such effects of synergy. So from what I could see this year, we could just interplant 2 cultivars. It would be enough to increase the yield. But there was, depending on the cross-pollen donor, the effect was not the same. some cross-pollen donors had higher effects on the berry weight, for instance, than other cross-pollen donors. So to maximize the fruit weight and the effect on of cross-pollination on fruit quality, it should be the best would be to choose carefully what cross-pollen donor to interplant with a given cultivar.
Is it? I seem to remember something about the relatedness of cultivars. I imagine some cultivars are quite closely related.
Yes, it was the hypothesis that I wanted to test this year, because it is what I said before. The berries are heavier because they have more mature seeds because of early acting inbreeding depression. So because of that, I thought that if the cross-pollen donor was closer to the recipient cultivar, I thought that the effect would be less, and so it would produce less mature seeds compared to another cultivar, another cross-pollen donor, which would be further in terms of genetics to the recipient cultivar. But finally, for now, I didn't find that there was an effect of the genetic distance between two cultivars.
This is fascinating work, and I guess the where a grower would see this come into place, you've already hinted at this, is typically today growers plant solid blocks of the same cultivar, and I imagine the immediate direct implications of this work is to, I don't know, maybe around field planting. Is that sort of would that be the main focus in terms of how future blocks are planted?
Yeah, I think that growers finally they do not interplant cultivars because blueberry do not need cross-pollination to set fruits. So we have quite much yields even without cross-pollination. But cross-pollination can increase the berry weight and so the yields and other variables of fruit quality. But as I said before, it depends also on the resources of the plants. So perhaps we cannot see the effect all the time, probably it is the case. And so I think that is why quite many growers don't see the effect of cross-pollination. For instance, I don't know, but if in the future we want to sell heavier berries, because I don't know the consumers prefer bigger berries. Perhaps it would be good to not pollinate all the flowers in a field, so to have this effect of cross pollination. It's just an idea. I could also see this year that the cross pollination increased to the fruit size between 10 and 100%, depending on the cultivar.
My goodness.
It decreased the time to ripen between 5 and 15 days.
Wow.
It also increased the firmness by quite 10% for each cultivar. And for now, I just study it in one cultivar, but I will process, I will measure the sugar and the acid content in the other cultivars. But for now, I only have one cultivar. And for this cultivar, the cross-pollination improved as well the sugar content, and it decreased the acid content, and it increased the Brix acid ratio. And I know that this variable are also quite important for the consumers. So the cross-pollination not only increased the fruit size, but it also have an effect on firmness and other variables of food quality, such as firmness and sugar and acid content.
All those things that make a high quality berry, a firm, well balanced flavor. So I love the way that you explain this, because in other systems you just don't get fruit without a pollinizer, and so you could see why very early on growers were aware of this. But here you still get the fruit, but what might be invisible to the growers, and this research should make clear, is that there's a whole lot of other benefits in terms of not only yield benefits but quality benefits that may come from actually adopting some other cultivars in their planting blocks.
Yes, exactly. The cultivar have an effect on firmness and sugar and so on. And so that's why I know that, for instance, in here at University of Florida, we have a breeding lab for blueberry, and they breed for better berries for the consumers. So the cultivar has an effect on it, but I could also see that the pollination has also an effect on it. The effect is less than the breeding, but for a given cultivar, we can improve the food quality because of pollination.
I suppose now you're going to be working on the northern cultivars. Do you have any expectations that this same pattern that you saw in the southern cultivars will hold when you turn your attention to the northern cultivars?
Yes, I expect exactly the same results, because it is the same genus. It is Vaccinium, northern and southern cultivars. And it is also the same species, at least one, between the two types of cultivars. So I expect exactly the same kind of results.
Well, thank you so much for that research update. Let's take a quick break. I have a couple more questions I ask all my guests. I'm looking forward to your answers. Welcome back. Okay, so we have three questions that I ask, most of my guests, but I'm really curious. Do you have a book recommendation for our listeners?
Yes, the book Nectar and Nectaries, written by Nicholson, Nepi, and Passini in 2007, from the edition Springer.
And why do you recommend that book? What's special about it?
Yes, it is just I love the topic of nectar, because for me it's like the hidden side of plant insect interactions. Most of the researchers currently they focus on the effect of landscape on wild bees and honeybees, because the landscape can bring some resources for the bees. But few researchers measure the amount of nectar secreted by the plants. Why it is very important? Because why the bees are attracted by the flowers? It is because of pollen and nectar, and in return, so they make the pollination. So that's why I'm particularly interested by the nectar, and in this book, we can learn many things, like the evolution of the nectaries, why they appear. And for instance, I could learn that the nectaries in the flowering plants, they appeared many times and they disappeared.
Really?
In many lineages. Yes, it's not appeared once and then all of the flowering plants that belong to this lineage produce nectar, then it could disappear. For instance, with the family Poaceae. So it is the rice, the
grasses. Yeah.
All the grasses. They don't produce nectar, but their ancestors produce nectar. So for this family, it disappeared with time. So I found it very interesting, and I found another example. So it is a researcher in Chile, if I remember well. So he worked in the archipelago Juan Fernandez, this close to Chile, and he found some flowers that produced nectar, but they were pollinated only by wind, because the ancestors of the flowers on the continents, these flowers produced nectar and they were pollinated by insects. But then on these islands, they lost their pollinators. The pollinators were not on the islands, but they continue to produce nectar. So we can see here that the secretion nectar is not mandatorily linked to insect pollination. And I found this example very interesting. Because with nectar, I like this topic because for me it can link the growers with the beekeepers. And for instance, in France, I worked on sunflower, and in France, some beekeepers thought that the new cultivars of sunflower produce less nectar than the previous one.
I've heard the same with oilseed rape, beekeepers complaining the same way.
In USA?
Yeah.
Okay.
Okay, but what did you find?
Okay. And the beekeepers thought it was because of the breeding. But so I measured the amount of sugar secreted between cultivars, and I found that there are differences between cultivars of nectar secretion. But I compared these amounts with the literature data, and I found no difference between the previous cultivars in the literature and the current cultivars.
Interesting.
And so there is no decrease of nectar secretion with the history of breeding.
Yeah, there's not a trend towards lower nectar. There's some cultivars that are high, some that are low, but there's not a historical trend.
Exactly. And I also tested if there was a correlation, a negative correlation between between the self-fertility rate of cultivars and the amount of nectar secreted. So we know that in sunflower, some cultivars need more pollination by insects than others. And I wanted to see if these cultivars secrete more nectar to attract more bees than the others. But finally, there is no correlation at all. So even if a cultivar do need less pollination by insects, it continues to secrete the same amount of nectar as the others.
Huh.
And I think it's because it's only human selection, it's not natural selection. I think the plants can lose their nectaries, but only with the time and with natural selection. In human selection, we don't select to have less nectar in plants. So that's why I think it can explain this discovery.
Well, I've always, I remember when I was back at the University of Calgary, there was an ongoing debate over whether nectar or pollen was a more expensive resource for a plant to produce. And, and I remember there was two kind of angles on this question. But these examples sort of suggest that maybe nectar, perhaps isn't the most expensive resource. It can stay. It doesn't get selected out. There's not the selection pressure isn't so driven towards eliminating it.
Yeah, I agree. Producing nectar can be a cost for the plants, but it depends on the strength of the selection pressure. So, and I think in our case, as we breed for cultivars, it cannot appear this selection pressure. That's what I think. And the other thing is that there could be an effect between there would be a link between the fertility rate and the nectar secretion if the two genes coding for these two phenotypes would be close on the same chromosome.
Mm-hmm.
If the two genes coding for these two traits would be close on the same chromosome, so there could be selected on the same time. They could be selected together.
Mm-hmm.
But if it is not the case, it can be completely independent, and that's what I think. That explains why I'm not very clear.
That those traits are discoupled.
Sorry?
The traits are discoupled. They're not. They don't kind of go. They don't track each other.
Yeah, they could be linked only if they were too close to each other on the same chromosome. If it's not the case, there is no special reason why they would be selected together.
Yeah, no, it makes sense, and I think that's that is maybe the where it would occur. It's being dragged along with some other trait that a breeder is selecting on. But otherwise, there's no reason for it. I've sometimes wondered if it just uses too much nitrogen and you're selecting for, higher efficiencies, if that would, but it's linked with so many different processes. It's not that simple.
I think so. Yeah, I think so. I agree.
Well, that was fascinating. That was an episode in its own right. And I can't wait to hear what your answer to the next question is. So we ask our guests if they have a go-to tool, the kind of, you can't imagine working without this tool in your work.
Yeah, it would be exactly in the same way as for the previous question. The tool that I like to use is capillary glasses. To measure the nectar amount in flowers, because as I told you before, I love to work on it. And I forgot to mention that for me, working on nectar secretion, as I work in crop pollination, working on this topic, I would like to answer the question of how many hives the beekeeper can bring in a crop, the maximum colonies he can bring in a crop, so that his honeybees will not competing with each other. So if we want to maximize the honey yields, there would be a limit above which the honey yields will decrease overall, will decrease for each colony. Mm-hmm. So as I work to know how many colonies we should bring to avoid any pollination deficits in crops. Mm-hmm. But there is also the question of
maximizing yield for the beekeeper.
Exactly. So that's why I am especially interested in the question of nectar.
It's a good question, because in some ways pollination rates are not, are kind of arbitrary. You think the pollination rate should be set at here's the amount where I no longer am making honey, and any stocking rate above this, or you're getting more yield, and that's what I should get paid. Like that, that seems a more sensible, because there's a stocking rate where the beekeeper maximizes their income, and the grower wants them to bring colonies in at a higher rate than that. And if you knew what that base rate was, then you could sort of calculate the differential or something.
Yes.
That's a great way you put it that way, because that is the kind of central question. They aren't the same stocking rates sometimes.
And I know that in France, as I mentioned before, beekeepers think that they produce less yields because of the cultivars. But the question of how many colonies are brought in the crops, we never know it. So perhaps sometimes it is because there are too many colonies in the same area. And perhaps it can explain, at least in parts, the decrease in honey yields.
Mm-hmm.
And I could never see any paper on this, so I am particularly interested to treat this question at some point.
Well, it does bring us to your go-to tool, the capillary tube, as a way to take nectar out of a flower and put it on a on a refractometer to get sugar content.
Yeah, exactly. And with this, so we could we can know the amount of nectar secreted by one flower. If we know the total number of flowers in a field, so we can make a prediction of the total amount of nectar secreted, and so we can make prediction on the yields. So yeah, this tool could help us for that.
I always wonder, just on this question, some flower, we've done a little bit of this work here, and some flowers are easy to get nectar with the capillary tube, and some, like, I don't know, alfalfa, or, a legume with many tiny flowers, or a composite, aren't so easy. Do you have any tricks for getting nectar out of hard to get plants with hard to get at their florets?
Yeah, there are many types of capillary glasses, and some of them are very, very tiny.
Oh, are they?
So, yeah, we can measure with it only 0.1 microliter. So they are very tiny. So with this we can probe tiny flowers. But I know that there is another method with very small flowers. We have to wash the nectar with the water, and then we take this water and then we measure the sugar content, and we can, if we know the volume of water we added, so we can know what was the amount of nectar at the beginning, of the amount of sugar. And so by this way, we can also measure the sugar in very small flowers.
Fantastic. All right. Well, our last question is, do you have a favorite pollinator species? Is there a species that you in particular find interesting or puzzling, or tell us your species?
Yes. I will not be very original for this question. My favorite species is Apis mellifera, the honeybee, because currently for crop pollination, the trends is for wild bees. In most of papers that I can read currently, they focus on wild pollinators. While for crop pollination, we need both wild bees and honeybees. Because the crops are not natural in a field. In a field, we have thousands and millions of flowers, and so even if we have many semi-natural habitats around the field, probably we would never have enough wild bees to not have pollination deficits. So I guess every time we will need honeybees, at least a few perhaps not too many honeybees, but at least a few colonies to not have pollination deficits. And so currently, I find that not many researchers work on honeybee management to increase crop pollination.
Oh, yeah.
So
you have to go back to the 80s and 90s to see that research. It's old now.
I really agree. There are few researchers currently, but not many. And so that's why I say that my favorite pollinator is this species because of that.
Excellent answer. Well, I'm really looking forward to your research this winter, and just fascinating conversation today. Thank you so much for being our guest.
Thank you, Anthony.
Thank you so much for listening. The show is produced by Quinn Sin and Neal, who's a student here at OSU in the New Media Communications program, and the show wouldn't even be possible without the support of the Oregon Legislature, the Foundation for Food and Agricultural Research, and Western SARE. Show notes with links mentioned on each episode are available on the website, which is at pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's several ways to connect with me. The first one is you can visit the website and leave an episode-specific comment. You can suggest a future guest or topic, or ask a question that could be featured in a future episode. But you can do the same things on Twitter, Instagram, or Facebook by visiting the Oregon Bee Project. Thanks so much for listening, and see you next week.